Hello everyone, my name is Heman Dangi and we are going to discuss about blood grouping. But before moving to our topic, let me first introduce you to the basic terms and concepts that will help us in the proper recitation of our topic. So, what is blood? The blood is the fluid connective tissue circulating in the different parts of our body. It plays a very vital role in our body. Let us discuss about the functions of blood. The most important function of blood is the transportation of oxygen and carbon dioxide. The blood transport oxygen from pulmonary level to different cells of our body in the form of oxyhemoglobin. Similarly, the blood transport carbon dioxide from the different cells to pulmonary level in the form of carboxyhemoglobin. Apart from this, the blood performs several messenger functions, including the transport of hormones and the signaling of tissue damage. The blood helps in the regulation of the body temperature. The blood performs several kind of immunological functions, like the cells present in the blood like neutrophils are phagocytic in nature. Now let us discuss about the components of blood. So the blood can be divided into two major parts: the plasma, which constitutes 55%, and the cellular part, which constitutes 45%. The out of the total plasma, 92% is water and remaining 88% is the dissolved substances. The dissolved substances include salts, foods like glucose and amino acids, various kinds of hormones, proteins like fibrinogen and albumin, and waste products like carbon dioxide and urea. The cellular part of the blood includes three primary types of cell: the red blood cell, white blood cell, and the platelets. The red blood cells are also called erythrocytes. They are biconcave in shape. They are enucleated and they consist of the red colored pigment which is called hemoglobin. The life cycle of red blood cell is approximately 120 days. The next type of cell is the white blood cell. The white blood cell is also called the leukocytes. The white blood cell can be divided into two categories: the phagocytes and the lymphocytes. The phagocyte ingest the microorganisms. The lymphocytes is of two types: the B lymphocytes, which produce the antibodies, and the T lymphocyte, which kills the infected and cancerous cells. The third type of cell is the platelets. The platelets are also called the thrombocytes and they help in the clotting of blood. Now let us discuss about the blood grouping. The term blood grouping refers to the entire blood group system comprising the red blood cell antigens whose specificity is controlled by a series of genes which can be allelic or linked very closely on the same chromosome. Blood type refers to a specific pattern of reaction to testing antisera within a given system. Over a period of time, our understanding on blood groups has evolved to encompass not only the transfusion related problems but also the specific disease association with RBC and surface antigens. Now, let us discuss about the blood group system. So, there is a society called International Society of Blood Transfusion which has recently recognized 33 blood group systems. Apart from ABO and rhesus system, there are many other types of antigens have been noticed on the red cell of RBC membranes. The first type of blood grouping system is the MNS antigen system. It was first described by Landsteiner and Levine in 1927. It is controlled by two types of genes. These are glycophorin A and glycophorin B. The blood group is used under control of an autosomal locus on chromosome number 4 and also under the control of a pair of codominant alleles LM and LN. Anti-M and anti-N antibodies are usually IgM types and rarely associated with transfusion reactions. The second type of blood grouping system is RH system. The RH is so called because it was first discovered in the monkey named rhesus monkey. If the red blood cell has Rh factor, then it is represented with a positive sign, and if it does not have, then it is represented with the negative sign. There is a specific disease which is associated with Rh factor. If mother is Rh negative and father is Rh positive, then the offspring will be Rh positive. In this case, first pregnancy is completely successful, but at the time of the first delivery, Rh antibody is formed in the mother's blood due to damaged blood vessel. So, in the next pregnancy, death of fetus will occur in the earlier stage due to the agglutination of the blood cell. This disease is called the erythroblastosis fetalis. The next type of blood grouping system is the ABO blood grouping system. It is the most important among all. According to the ABO blood grouping system, there are four types of blood groups: blood group A, blood group B, blood group O, and blood group AB. The ABO blood grouping system was given by Karl Landsteiner in 1901. He, for the first time, established that the serum of one individual would clump the cells of another individual. This is because the cells contain a substance known as antigen and the serum contain the antibodies. The table which is shown in the slide clearly shows that a person with blood group A would have the antigen A and the antibodies in the plasma would be anti-B. The person with blood group B would have the antigen B and the antibodies in the plasma would be anti-A. Similarly, the person with blood group AB would have both the type of antigens A as well as B, but in this case there would be no antibodies in the plasma. The person with blood group O would have neither of the antigens and both the antibodies will be present in the plasma. It is very clear that the person with blood group A is the universal donor and the person with blood group AB is the universal acceptor. Now let us discuss about the genetic basis of ABO blood grouping. So the ABO blood grouping is controlled by a gene called I gene. The I gene stands for isoagglutinogen. There are three different allelic forms of this I gene: the IA, the IB, and the small I. This this is an example of a very important concept of genetics which is called multiple allelism. It means that three different kinds of alleles, that is IA, IB, and small I, are governing the same character, which is the ABO blood grouping. The IA and IB produce slightly different form of sugar, while allele small I does not produce any kind of sugar because the human beings are diploid organisms. It is very clear that IA and IB are completely dominant over small I. In the other words, if IA and small I are present, only IA will express itself. Similarly, when IB and small I are present, only IB will express itself. But when IA and IB both are present together, then both of them will show their own types of sugar. This is because of the phenomenon of codominance. Codominance is the heterozygous condition in which both the alleles at a gene locus are fully expressed in the phenotype. Hence, the red blood cells will have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible. These combinations are shown in the table. It is very clear from the table that if parent 1 will have allele A and parent 2 is having allele B, then the genotype of the offspring is IA and the blood group will be A. If the parent 1 is having allele IA and the parent 2 is having allele IB, then the genotype of the offspring will be IAIB and the blood group of the offspring will be AB. Now let us discuss about the Bombay phenotype. The Bombay phenotype is so called because it was for the first time discovered in the Bombay. It is the city in the Maharashtra state of India. Persons with rare Bombay phenotype are homozygous of H gene, which is the precursor of ABO blood group antigens. They do not express H antigen on their RBCs. As H antigen acts as a precursor, its absence means the absence of antigen A and B. However, the individuals produce isoantibodies to H antigen as well as antigens A and B. Now, let us discuss about the experimental procedure by which the ABO blood group is determined. In this experiment, we will take a clean glass slide and mark 3 different points as anti-A, anti-B and anti-D. Now we will put the blood drops at the 3 different points carefully. Then we will carefully add the anti-sera A, anti-sera B and anti-sera D at the respective places. Then we will mix the anti-sera with the blood with the help of clean needle. But it is very important to note here that for the mixing purpose we will use 3 different needles. We will not use the single needle for more than one spot. Then we will leave the sample for 2 minutes and we will note the observation. It is very important to note that the agglutination shows the positive result. In the picture which is shown in the slide, the A spot does not show any kind of agglutination while B and D spot shows agglutination. It means the blood group will be B positive. In case of blood group O, neither A nor B shows the agglutination. In the last, I would like to discuss about the significance of the blood grouping. So, the very first significance is the importance of blood grouping in the transfusion. So, whenever the doctor prescribes for the transfusion of blood, the very first thing that he asks us is the blood group of ours. So it is very important to transfuse. It is very important to note the compatibility of the blood before its transfusion. If we transfuse the incompatible blood in a person, then it will lead to the clumping of the intravenous blood and it can be fatal. The second most important significance of blood grouping is the paternity disputes. The blood grouping plays a very vital role in the paternity disputes. The blood group help us in knowing the DNA sequences of the person whose body is found in the particular kind of criminal case. As we know that red blood cells has no nucleus and white blood cells consist of nucleus. So we can extract the DNA from the white blood cells and we can perform the DNA barcoding of that particular person. The next important aspect of the blood grouping is the medico-legal use. As we know, every kind of physical assault involves blood as an important physical evidence. So, blood can be very helpful in knowing the true mystery of the cases. The next important aspect is the association with the susceptibility to disease. In the last, the importance of blood grouping is during the pregnancy. It is also very important. Thank you so much.